So how do you feel about drug reprofiling or repurposing rather than, sitting in the lab and cooking up a new drug? What do you think is a better approach?
I think both are required. I don't
Okay.
think you want to go with one or the other. New molecules are going to have potential new attributes, so you don't want to not do that. But that said, do we know everything about the molecules we've already invented? So there are drugs out there. I mean, you can think of a whole bunch of molecules out there that started off with a particular target and are not being used for that target. I mean, Viagra is a classic example of that.
Definitely.
They were targeted for coronary artery disease. They can deal with people in a way, because this drug was supposedly targeting the vasodilatory mechanisms in the heart. Oops, it didn't work very well there. Meanwhile, they found out in the dog experiments that the dogs were getting erectile responses. Like, okay, so what's going on?
Okay.
So the repurposing of a molecule with a known safety profile is a very smart thing to do. In fact, you may not remember this, but if you go back in the literature, you're seeing, this is probably 15 years ago or longer, there were a bunch of drugs on the market. They were calcium channel blockers, and they were used for things like hypertension. They were used for coronary artery disease or congestive heart failure. But these drugs were very short-lived. So
Okay.
they quick in, quick out, and as a result, there was a side effect profiles sufficient that there was morbidity and mortality associated with taking these drugs. So Dear Doctor letter goes out in Canada to stop people from taking these short-acting drugs, because all of a sudden this was discovered, and as soon as it was discovered, Health Canada said, we're going to pull them off the market, but we have to have other ones ready.
Okay.
So the companies repurposed the same drugs and created sustained release formulations, because it wasn't known until post-marketing surveillance revealed that the drug was creating these adverse effects in a small percentage of patients, and they could resolve them by changing the pharmacokinetics of the drugs.
Gotcha.
No longer oscillating the concentration to the drug, keeping them stable, preventing the body from having a maladaptive response to the oscillation so that there was a problem. So there's a repurposing example. Pharmacokinetics is a very important feature, and don't forget it when you think of a new molecule.
Okay.
Yeah.
So you think that drug profiling can definitely, or drug reprofiling, sorry, can save people a lot of money when it comes to creating a drug. So say like Viagra, if it was intended for one thing, didn't work out, but you use it for something else, that can save you a lot of money when it comes to like investors
Yep.
and the companies that have stakes.
Absolutely, absolutely. And these things are extremely expensive. So you imagine TAP, they never got back their money. Right? Really, they spent a lot of time and energy, even if they're making $150 million a year on it.
Yeah.
That's not enough to pay back the bills. That basically pays the costs of doing it at that point.
Okay.
So it's, how do you get back the other $300 to $400 million you spent on the drug development? So they move on to another drug and they leave it behind and they never repurpose it. So there's an example of, wow, if the mentality was always to repurpose or examine what you have and say, is there something we can do better? If that mentality was always there, then I think we'd probably be farther ahead with a lot of drugs. Right now, it's new molecule, new chemical entity, right?
Yes.
Easier to patent a new chemical entity, right? NCE is called, right? The short form NCE. Because new chemical entities are going to be big blockbuster, hang on, with no known safety profile.
Yeah.
So you go through all that and then you're killed by your safety profile. So to me it's very much a balance, but people should always do both. The companies should examine all drugs available to them in any way possible and have a team of people doing it, and the reprofiling is, to me, a key part of it.
Okay. And so the drug that you brought to market, were there initially problems with pharmacokinetics or pharmacodynamics, ADME, toxicity? How was the journey, kinking all the bugs out?
Trial and error. It was known to have a fast kinetic profile, so
Okay.
it was going to be chewed up metabolically in the body quickly, so you had to give it in a way. No one really knew how to give it, so we did both animal and human experiments. We actually had patients treated and examined them, but obviously this is a drug that's been used before, so it was not like we were repurposing it, we were reprofiling it.
Okay.
So we had to determine what was a good profile, what was a bad profile, and that good profile ended up being one where you did not want a fast peak. The faster onset was a recipe for disaster.
Okay.
Because the body couldn't handle fast triggering of dopaminergic receptors in a non-selective manner, because it would offset things like create syncope, dizziness. Because the body, you were triggering, erectile responses are cardiovascular responses.
That's true.
So in this case, it was triggering cardiovascular responses that you did not want. So what if you had a slow onset, and literally over 15 to 20 minutes as opposed to over 3 minutes? You generated a much more selective response when in the face of a sexual response or a sexual stimulation, and it ended up being a much more selective signal in our hands until they made a mistake and made it too fast an onset.
Oh, okay.
So they made a mistake in not realizing the key part of the pharmacokinetics, which we were explaining to them as often as we could, but it was out of our hands. We'd licensed it, right? We were consulting and advising,
Okay.
but we'd licensed it to them.
All right. So complete control was out of your hands at that point?
That's correct.
Okay. So to bring a successful drug to market, where exactly do animal models fit in? So at what point is a drug ready to bring to animal models, and what considerations go in, with regards to dosing?
Yeah, that's a hard one. Scaling from animals is a tough one, but understanding, like a rat, pretty well everything that I do now is rat related because I can recapitulate most cardiovascular responses in rats very readily. So I can understand the role of the system, if you want, the involvement of multiple systems, it does that pretty well. When you're trying to look at anything I've done, almost all the studies were generated initially in the animal studies. We examined the possibility, we looked at the scope, we looked at concentration response relationships and dose response relationship, then moved into the human studies, scaled it appropriately, and we're within less than a third of an order of magnitude right from the get-go. So
Wow.
it wasn't that hard to do. So you can imagine a lot of our patents had scaling issues in it, and we had zero problems. Once we learned what we learned,
Okay.
we had example after example, and our patents showed that we were experts in scaling. We knew how to scale an animal study to a human study, and we were right. So yeah, you can't do some of the experiments that you want to do with humans. The variation in human beings, you're getting treatable humans with erectile dysfunction who are maybe 50 to 60 or 70 years of age. So they've got other illnesses. They could have some diabetes, they could have anxiety, they could have relationship issues,
All right.
on and on. So to control all of those while you're testing for a drug is not trivial.
Okay.
So a rat gives you a much cleaner background where you're examining the biological potential for
Okay.
it. You may not get the exact translational aspect in terms of the diversity of patients you're going to face, but at least you understand what the biology is going to do.
Okay.
And that's step one.
So what do you think are the limitations of using animal models? So other than the fact that humans have a lot of variability in terms of genotype and phenotype and other social stressors that happen, do you think there are any types of drugs that have specific targets that aren't good to test in rats and mice?
That's too broad a statement to say they're not good. You have to be careful with the model. You have to be, I was on the proof of principle panel for Partech for about 5 years, where we gave out money to people who were exactly doing that. They were trying to scale their basic science into the translational world, and I was a bit of a bear, I guess, for demanding that they knew that their model was the right model to do that in. And if somebody was just doing an animal model for the sake of doing an animal model, I would take them to task, because if they didn't understand what they were doing, I'm sorry, they're wasting time and energy and money.
Okay.
So it does matter. You have to know a lot about it, and it's one of the things that I think has happened. Not enough training of people who are really understanding of whole animal, whole organism situations are being trained. Everybody works in that way in my lab, but those are tough slogging experiments to do. They're scaled quite high. We have some projects have 7 to 8 people on them, right, to work on those, and they're longitudinal. But I mean, working in a single nude mouse only, and doing a cancer study only in that, and maybe adding in vitro stuff as well, is not sufficient to scale to a human. It's not. It does not add in all the difficulties of a cancer condition. That's a tough one. Those are the kind of issues. That said, it tells you whether the target has got the possibilities of it, but the rest of it is a lot of work. Yeah.
So were there any phases in clinical trials where you were scared that your drug wouldn't pass and maybe just shut down completely? Were there any moments like that?
Oh, every day. You've got this opportunity for success, right? You can imagine if with our endothelin story, we were telling people that their drugs were going to fail. I didn't think I hoped for it, but when the failure after failure came down the pike, and I said, well, that's what we told you. Our experiments in animals predicted the failure in humans.
Okay.
So there's an example of the animals were exactly the window. When you looked at the rat studies on the centrally acting erectogenic agent, and then you look at the successes in some of the human trials, and then the later not so as good a success as the formulation didn't work, you go like, oh, come on, this isn't fair. We know this works, you just have to get it right.
Okay.
So there's a lot of anguish around that, a lot of anguish. And
Alright.
probably the biggest frustration though was the fact that it didn't get used the proper way and could have expanded to the female condition as well.
So did you do anything different from the regular drug development processes, or what was being done by your peers or people around you at that time in terms of drug development that you felt helped you pass your drug?
Picking the right people to work with is probably the biggest deal of all.
Okay.
You pick the wrong people, recipe for disaster. Pick the right people who are good listeners, who've got a strategy, who have got some common sense and are willing to listen and adjust their trajectory. That we did that. I think we were happy with that in most of our choices, but there's so many stories of people not picking the right people to work with.